Welcome to SATORI, a system for ontology-guided visual exploration of biomedical data repositories. In this video we introduce SATORI and demonstrate 3 use cases how SATORI can aid exploring datasets. A dataset contains one or more data files associated with a set of metadata attributes like the file name, creation date, technology, organism, or disease state. Some attribute values are annotated with ontology terms, providing additional context or relationships. For example, when a file is associated with the term neuroblast, we can infer that it is also associated with the term neural stem cell, which is a parent of neuroblast. Stem cell, in turn, is a parent of neural stem cell. SATORI helps us to explore the repository with this information. Here we see the landing page. On the left side of the interface is the dataset view, consisting of the search input, filtering and sorting options, and a dataset list. Each dataset is represented by a snippet called surrogate, containing the title, sharing information, and the keyword and context phrase showing the matched keywords when a search is executed. A dataset view can be opened at any time by clicking on the title of a dataset. It contains the description, technology type, sample sources, analyses, references, and experimental protocols. On the right is the exploration view, consisting of 2 visualizations, the node link diagram in the top half and the tree map in the bottom half. The tree map visualizes the abundance of annotation terms across the whole repository as nested rectangles. The size of an annotation term is defined as the number of datasets that have been annotated with that term at least once. The color of a rectangle indicates the depth of the ontological subtree below this term. Nodes in light gray without a border symbolize terminal nodes. The node link diagram visualizes annotation terms as connected nodes. Links between nodes characterize parent-child relationships, where the parent represents a superclass of the child. For example, chemical component is a parent of biotin. Each node is superimposed with a bar and a line indicator. The bar represents precision of a given term, which is the number of currently retrieved datasets annotated with that term, divided by the total number of retrieved datasets. The superimposed vertical line indicates the recall of a term, which is the number of retrieved datasets annotated with that term, divided by the total number of datasets that have been annotated with that term across the repository. Here, recall is 1 for all terms, as we are looking at the entire repository. All 3 views are interlinked. Whenever the user hovers over a term in the tree map or node link diagram, the identical term in the other plot is highlighted. The datasets annotated with that term are also highlighted. In the same way, annotation terms in both plots are highlighted when the user hovers over a dataset. Depending on the task and personal preference, one of the 2 visualizations can be minimized to give more space to the other. Each component of SATORI features an interactive tutorial that can be opened by a click on the info buttons. For this demo, we have imported the Stem Cell Commons, a repository of microarray and RNA-seq datasets from studies related to stem cells. Assume we want to find all datasets related to leukocyte. Let's search for it. Nothing found. We might be disappointed, but keep exploring by native cell to check if we can find similar datasets. We see that the most prominent cell types are somatic, animal, and precursor cell. We are sure that leukocyte is not a precursor cell, so we exclude datasets related to it. We inspect the next column and find leukocyte with a precision higher than 0, so we query for leukocyte. We find 5 datasets. Since we want all leukocyte datasets, we have a quick look at the recall, which tells us how many more datasets related to leukocyte are available. We notice that we found less than half of all leukocyte-related datasets. Maybe some of the datasets related to leukocytes do contain other files related to precursor cells, which we excluded earlier. Since we queried for leukocyte anyway, we can remove previous queries. Now we see that the recall went up to 1, meaning that we found all datasets related to leukocytes. Next, we remove the query for native cell to explore which species are associated with the datasets we have found. It turns out that all of them are related to mouse samples. Interestingly, there are also cancer-related datasets. Since the title of the study does not provide much information, we open the dataset summary view. Here we see that all biological samples are subtypes of leukocyte, but nothing has been explicitly described as leukocyte, which is why we haven't found anything with a text-based search. Next, assume we are a data curator and interested in investigating the current state of curation, and if there is room for improvement. We start by examining the tree map. Since the first level of annotation terms looks fine, we increase the visible depth to find terminal nodes of high abundance. We spot biotin, which occurs relatively often, indicated by the size of its rectangle. Zooming into biotin shows that 134 out of 199 datasets are associated to it. Given that stem cell commons contains many microarray datasets, this overrepresentation seems reasonable. We go back and investigate assay types. We notice that only 195 out of 199 are annotated with assay. This seems surprising, so we decide to look at datasets that have not been annotated with assay by changing the query mode of assay to not. Using the dataset preview, we find out that all 4 datasets are missing the assay annotation. 3 of them could be annotated with epigenetic modification assay, and the other one with transcription profiling assay. We notice that the dataset description for assay type seems to be more precise than the ontological annotations. To confirm this, we remove the not query for assay and restrict the datasets to be related to transcription profiling assay. Since we know that biotin is an indirect description for microarrays, we highlight related datasets. Looking at the first couple of datasets, we confirm that the ontological annotations do not distinguish between RNA-seq and microarrays, which could be improved in the future. For the final demo, we have imported studies from metabolites, a growing collection of metabolomic datasets. Assume we are a project leader and want to learn about the content of metabolites in terms of disease-related studies, in particular cancer. We first have a look at the tree map to get an idea of the diversity of annotations. We notice that metabolites represents a diverse set of studies indicated by many small terminal nodes. We spot neoplasm in the lower half given its relative dark background and zoom into it. 3 datasets are retrieved, which we put in a data card for later comparison. We notice that the recall of the parent term disease is low, so we zoom out to find more disease-related datasets. In order to assess which species have been studied given the retrieved datasets, we zoom all the way out and query for disease instead. We decide to look at the eukaryotes and find that most datasets are associated with human and some with mouse. After querying for human, we add all of the retrieved datasets to the data card. Now we remove the query for disease and search for it instead, to assess non-ontological metadata as well. We find 20 datasets, of which some, but not all, overlap with the previously saved datasets, indicated by yellow stars. We add the newly found datasets to our data card for later discussion in the project meeting. Finally, out of curiosity, we clear the search for disease and look at the most commonly studied species in general. We notice that a large set of studies is related to green plants, which highlight the diversity of metabolites again. You can find out more about SATORI at satori.refinery-platform.org. Thank you for watching.